There is a small piece of information about yourself that, depending on who you ask, either means absolutely nothing or means everything. It's a letter and a symbol, a plus or a minus. It shows up on medical forms, blood donor cards, dog tags. Most people glance at it, file it away, and never think about it again. But a significant portion of the population has spent years, sometimes decades, convinced that their version of that symbol is evidence of something ancient, something hidden, something that science either can't explain or won't. We're talking about Rh negative blood, and the story around it is one of the strangest collisions of real biology, genuine scientific mystery, and full-blown mythology that exists anywhere on the internet today. Here's the thing though, the mythology didn't come from nowhere. That's what makes this worth taking seriously. The questions people ask about Rh negative blood. Why is it rarer? Where did it come from? Why does it cause problems during pregnancy? Why is it distributed so unevenly across the world's populations? Those are legitimate scientific questions, and some of them don't have clean, settled answers yet. The gap between what we know and what we don't is exactly where the stories rush in. And the stories that have rushed in are extraordinary. Ancient bloodlines, pre-human ancestors, extraterrestrial intervention, Nephilim, royal lineages stretching back before recorded history, hidden traits that mark the carriers as fundamentally different from the rest of humanity. If you have spent any time researching Rh negative blood online, you have encountered at least one of these, probably all of them. So here is what this is going to be. We're going to start with the science, because the science is actually fascinating on its own terms, and most of the mythology gets the science wrong in ways that matter. Then we're going to look at the history, the real migrations, the populations that carried this trait across continents, the moments in human history where blood type suddenly became medically urgent. Then we're going to look at the stories, the Basque origin theories, the royal bloodline claims, the alien intervention hypothesis, the scriptural interpretations. We're going to take them seriously enough to examine them carefully, which is the most honest thing you can do with an idea. And we're going to keep asking the question that actually matters. What does the evidence support? What is speculation? And what remains genuinely open? Before any of that, though, let's pause on something worth noticing. Blood type is almost unique among biological facts in how personal it feels. Your height, your eye colour, your general ancestry, these things feel like traits you wear on the outside. But blood type is invisible. It's interior, it runs through you. And that makes it feel like a secret, like something coded into you at a level deeper than appearance. That feeling is real even if what people build on top of it often isn't. It's why blood type personality systems became massive in Japan and South Korea. It's why people include their blood type in dating profiles in parts of Asia, the same way others list their zodiac sign. Blood feels like identity in a way that other biological markers don't quite manage. And when you add the word negative, when you frame it as an absence, something missing, something that most of the population has but you don't, that feeling intensifies. People who discover they are Rh negative often describe it as a moment, like finding out you belong to a category you didn't know existed. And human beings are pattern-seeking creatures. Once you know you're in a category, you want to know what the category means. You go looking for answers, and the answers you find depend entirely on where you look. The questions themselves are genuinely interesting. Why does Rh negative blood exist at all, if most of the human population carries a different version of the protein? Where is it most common? And why is it so dramatically concentrated in certain corners of the world, while being almost absent in others? Why does it create dangerous complications during pregnancy, in ways that most blood traits don't? And where exactly did it come from in our evolutionary history? That last one is the question with the most unresolved edges, and it's the one that has generated the most creative explanations. We'll get to all of it. The genetics, the anthropology, the migration patterns, the medical history. We'll get to the Basque people, who have the highest concentration of Rh-negative blood anywhere on Earth, and the theories about what that means for human prehistory. We'll get to the European royal families and the medical tragedy that ran through their bloodlines for generations. We'll get to the claims that this blood type literally shouldn't be in the human genome, and what the people making that claim actually mean by it, and whether the biology supports anything like that conclusion. But right now, before all of that, I want to know something. If you're listening to this right now, drop a comment and tell me where you are and what time it is. I genuinely check these. There is something remarkable about the fact that this script is playing simultaneously in living rooms and on commutes and through earbuds in the dark in cities I'll never visit, in time zones spread across the entire planet. Berlin at breakfast, Mumbai at midnight. Someone in New Zealand already into tomorrow. If that's you, say something. Tell me where you are. Because here's the thing about a mystery like this one. It connects people. The same questions about Rh-negative blood have been asked by a doctor in Sao Paulo, a historian in Scotland, a teenager who just got their blood type result back and typed it into a search engine at 2 in the morning. The mystery is shared even when the answers people find are very different. So let's start at the beginning. What is the Rh factor actually? Not what people say it is, what it actually is. Your blood is doing something right now that you've never had to think about. It's circulating, carrying oxygen, fighting off things that shouldn't be there, doing all the maintenance work that keeps you alive without any conscious input from you whatsoever. And on the surface of every red blood cell, there is a collection of proteins and antigens, molecular structures that your immune system uses to distinguish between what belongs in your body and what doesn't. Some of those antigens are incredibly important medically. Others are mostly irrelevant to your health on a day-to-day basis. The Rh factor falls somewhere in between, and understanding why requires backing up a little. Most people have heard of blood types. A, B, AB, O. These refer to the ABO system, which is about a specific set of antigens sitting on the surface of your red blood cells. Type A means you have A antigens. Type B means you have B antigens. Type AB means you have both. Type O means you have neither. Your immune system also produces antibodies against the types you don't have, which is why giving someone the wrong blood type in a transfusion can be fatal. Their immune system recognizes the foreign antigens and attacks. The ABO system is the one most people learn about first, and it's the one that gets matched for blood transfusions and organ transplants. But the ABO system is not the only antigen system in your blood, not even close. Scientists have identified over 40 distinct blood group systems containing more than 300 individual antigens. The Rh system is one of them, and it happens to be the most medically significant after ABO. The Rh system itself contains around 50 different antigens, which is where things start getting more complicated than the simple plus or minus label suggests. But the one that matters most, the one that the plus or minus is actually referring to, is called the D antigen, specifically the Rhd antigen. If you are Rh positive, that means your red blood cells carry the D antigen on their surface. If you are Rh negative, that means they don't. That's it. That's the whole thing the symbol on your donor card is describing. The presence or absence of one specific protein on the surface of your red blood cells. Now, the reason this matters medically is the same reason the ABO system matters. Your immune system is extremely good at detecting things that don't belong. If you are Rh negative and your bloodstream is exposed to Rh positive blood, your immune system can learn to recognize the D antigen as foreign and build antibodies against it. Those antibodies then become a problem if Rh positive blood enters your body again, because now your immune system knows exactly what it's targeting. This is why Rh matching matters in blood transfusions, and it's why it matters enormously in pregnancy, which we'll get into later. But here's the important thing to establish right now. because this is where a lot of the mythology goes sideways immediately. Being Rh negative is not a deficiency. It is not a mutation in the sense of something going wrong. It is not evidence that something was lost from your genome that should be there. The framing of absence, the minus sign, makes it sound like Rh negative people are missing something that Rh positive people have. But the actual genetic story is considerably more interesting than that. The D antigen is produced by a gene called Rhd. If you have a functional copy of this gene, your red blood cells produce the D antigen and you are Rh positive. If you carry a version where the Rhd gene is deleted, or has a variant that prevents the antigen from being expressed properly, your red blood cells don't display the D antigen and you are Rh negative. In most cases, in people of European ancestry specifically, Rh negative status results from a complete deletion of the Rhd gene. The gene is simply absent from that location on the chromosome. In some other populations, Rh negative status can result from different variants, point mutations or other changes that functionally silence the gene without removing it entirely. Which is already a hint that the genetics here are more varied and more geographically complex than the simple binary label implies. What this means is that Rh negative blood isn't a presence of something strange. It's the absence of a gene that produces a protein. And gene deletions, gene variants, these are normal features of human genetic diversity. They happen constantly throughout the genome. The question of whether the presence or absence of the Rhd gene confers any advantage or disadvantage has been studied, and the answers are nuanced and incomplete, which is a more honest way of saying we don't fully know. Some researchers have suggested possible links between Rh status and disease resistance or susceptibility. A few studies have looked at whether Rh negative individuals might have different responses to certain parasites or pathogens. None of this is settled. The most accurate statement right now is that being Rh negative appears to be a normal human variation that is largely neutral in terms of survival advantage, with the notable exception of the complications it creates in certain pregnancy scenarios. And that neutral variation framing matters, because one of the central claims in Rh negative mythology is that the trait is so unusual, so out of place in the human genome, that it requires a special explanation. Alien intervention, a separate ancestral lineage, something fundamentally different in the origin story of people who carry it. But the biology doesn't actually support the premise of that argument. Unusual in terms of frequency, yes. Out of place in the genome, not in any scientifically meaningful sense. There's nothing about the Rhd deletion that defies explanation through ordinary human evolution and population genetics. The mystery, to the extent there genuinely is one, is about distribution and timing, not about the gene itself being impossible or inexplicable. Now here's where things get genuinely interesting beyond the basics. The Rh system, as we mentioned, contains around 50 different antigens. What most people refer to as simply Rh positive or Rh negative is only the tip of that system. There are other antigens in the Rh family, labelled with letters and names like C, CE, E, and dozens more. These don't get the same cultural attention as the D antigen because they're less medically dramatic in most situations, but they are part of the same underlying system. And people who are considered RHS negative based on the D antigen can still carry these other Rh variants in all sorts of combinations. There's also the phenomenon of weak D and partial D, which are categories that exist precisely because the plus or minus binary is an oversimplification. Some individuals have a version of the Rhd gene that produces the D antigen, but in reduced quantities, or with a slightly altered structure. These people may test as Rh positive in some screening scenarios and Rh negative in others, depending on the sensitivity of the test being used. The clinical management of these individuals is more complex than the simple positive-negative framework allows for, and it has real consequences in medical settings. The point is that the Urich system, like most biological systems, is considerably more layered than the version that shows up on a blood donor card. The simplification is medically useful because the D antigen is the clinically significant one in most situations. But it has also created a cultural story in which RIH positive and RIH negative are treated as two fundamentally distinct categories of human being, when the reality is closer to a spectrum of variation in a single gene system, distributed unevenly across populations for reasons that relate to migration, genetic drift, population bottlenecks, and possibly selection pressures that we haven't fully characterized yet. That uneven distribution is real. It's documented. It's one of the legitimately puzzling things about this trait. Why is Rh negative blood so dramatically concentrated in certain parts of the world and so vanishingly rare in others? That's a question with real scientific content, but the path to understanding it runs through population genetics and human prehistory, not through claims about alien intervention or bloodlines that bypass the human evolutionary tree entirely. The science here is actually rich enough that it doesn't need embellishment. The RHD gene, its deletion pattern, the way that deletion is distributed across the globe following the paths of ancient human migrations, the way different populations develop different mechanisms for producing the same negative phenotype. This is a genuine puzzle with pieces that are still being assembled. Some of those pieces are unexpected. Some of them raise questions that researchers haven't resolved. The mystery is real. It's just a different kind of mystery than the one that gets 1 million views on YouTube, which, depending on your appetite for actual complexity, might be more interesting or less. Either way, understanding what the Rh factor actually is, at the level of genes and proteins and immune response, is the only foundation from which any of the more ambitious questions can be honestly examined. Because if you get the biology wrong at the start, everything you build on top of it is going to be wrong in the same direction, and probably in a direction that feels more satisfying than it should. So now that we have the basic mechanics, let's talk about how this trait actually gets passed down, and what the patterns of inheritance reveal about where Rh negative blood came from and why it persists. If you've ever compared blood types in your family, you've probably seen something that looks a little weird. 2 Rh positive parents have an Rh negative child. One sibling is negative, the others are positive. A grandparent was negative, none of their kids are, and then suddenly the trait pops up again in the grandchildren. To people used to thinking in straight lines, that can feel like a glitch in the matrix. But this is exactly how you'd expect Rh to behave once you understand the rules it's playing by. Rh status, at least in the simplified form of positive and negative, follows a basic recessive inheritance pattern. It's not the only thing involved in the Rh system, but for the D antigen, the logic is pretty straightforward. Think of the RHD gene as coming in 2 main versions for our purposes. One version we'll call Big D, the functional gene that produces the D antigen. The other we'll call Little d, the deleted or non-functional version that doesn't. You get 1 copy from your mother, 1 from your father. If you have at least 1 Big D, your cells make the D antigen and you're Rh positive. So someone who is DD or DD will test positive. Only if you have 2 copies of little d, so DD, will you be Rh negative. That's what recessive means here. The negative trait only shows up when both gene copies are the non-functional version. Now plug this back into the family patterns that confuse people. 2 Rh positive parents could both be carrying 1 Big D and 1 little d without knowing it. On paper, they're both positive. Genetically, they're what's called heterozygous. Each of their children has a 25% chance of inheriting little d from both parents, ending up DD and being Rh negative. No mutation needed, no secret ancestry, just the math of recessive inheritance. The reverse pattern makes sense too. An Rh negative person is, by definition, do do. They can only pass little d to their children. If they have kids with someone who is DD, all of those children will be DD. Every one of them will be Rh positive, even though every one of them is carrying the negative allele. From the outside, it looks like the trait disappeared in that generation. In reality, it's just hiding in plain sight, waiting for the right combination of partners in a later generation to produce a negative child again. This is why family stories built around our blood is different can get misleading very fast. A Rh negative can look like it suddenly appears in a lineage or suddenly vanishes, not because anything mysterious is happening, but because recessive traits are very good at skipping visible generations while quietly riding along in the genome. Zoom out from families to entire populations, and the same basic logic still applies, just summed over millions of people. Geneticists use something called Hardy-Weinberg equilibrium as a mental baseline for this. It's not a law in the sense of gravity, it's more like the frictionless surface in physics problems, an idealized situation that lets you see what would happen if nothing interesting was pushing things around. The idea goes like this. Imagine a big population where people are randomly choosing mates. Nobody's migrating in or out. No one gene version gives you a survival or fertility advantage. And the population is large enough that chance fluctuations mostly cancel out. Under those conditions, the frequencies of gene variants, alleles, settle into stable proportions. If the negative allele, little d, sits at, say, 40% in that theoretical population, and the positive allele, big D, sits at 60%, then you can predict how many people will be D, 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 and D. The math tells you what percentage will be visibly Rh positive, and what percentage will be Rh negative, and those proportions will stay the same generation after generation, as long as the assumptions hold. Real human populations, of course, are almost never living in that ideal world. People move, groups split and rejoin. Wars happen. Plagues happen. Certain traits become a little more or a little less advantageous in particular environments. But Hardy-Weinberg gives you a baseline. It tells you that if you see allele frequencies drifting around, something is doing the pushing, and that something doesn't have to be dramatic. It doesn't have to be aliens or secret hybrids or coded interventions. In fact, one of the most powerful forces in population genetics is incredibly boring. Tiny differences in survival or fertility stretched out over long periods of time. Imagine that, in a particular environment, people with a certain genotype have, on average, 2.5 surviving children instead of 2.0. That's not a number you or anyone around you would notice. It's noise at the level of individual families. But played out over hundreds of generations, those extra 0.5 surviving children per couple become very real. The allele that slightly nudges survival or fertility upward can slowly climb from rare to common, or from common to dominant, without anyone in that population ever feeling like a revolution just happened in their genome. Conversely, even without any survival advantage or disadvantage, allele frequencies can wander just because of chance. This is genetic drift. In a small population, random events, who happens to have more children, who happens to die young, can shift allele frequencies dramatically in just a few generations. No selection pressure required. Now add one more ingredient, founder effects. Picture a small group leaving a larger population and starting a new one somewhere else. An island, a valley, a mountain refuge. The genetic makeup of that founding group is, by definition, a sample. It may not perfectly represent the larger group they came from. If, just by chance, a few of those founders happen to carry more copies of the Rh negative allele than average, their descendants will inherit that skewed starting point. Over time, the new population can end up with a much higher Rh negative frequency than the parent population, even if nothing about their new environment particularly favours or disfavours Rh negative. This is one of the main ways you get odd-looking pockets of genetic traits. isolated groups founded by a small number of people whose genes, by luck, weren't a perfect match to the parent population. Combine founder effects with genetic drift in a group that stays relatively small and isolated, and you can turn a minor allele into a local hallmark trait in just a few thousand years. Then, later, migration happens. People who were once isolated start moving, trading, marrying into neighbouring groups. Their genetic signatures begin to blend. Areas that used to have almost no Rh negative may see it introduced as carriers move in. Areas that had high frequencies may see them diluted as more Rh positive genes enter the mix. None of this requires a single special event. It's the same quiet arithmetic of genes and reproduction applied over geography and time. When you put these pieces together, recessive inheritance, Hardy-Weinberg as a baseline, small differences in survival, founder effects, drift, and migration, you get a toolkit that can explain a huge amount of the patterning we see in Rh-negative distributions. Not every detail is nailed down. There are still debates about whether Rh status has had any meaningful selection pressures in particular environments. But you don't have to reach outside known biology to understand why some families and some regions have much more Rh negative than others. This matters because a lot of the wilder claims about Rh negative start from a false premise, that its distribution is so strange it must be evidence of something fundamentally non-human going on. Once you understand how genes actually move through populations, that claim loses most of its force. The real questions become more specific and more grounded. When and where did the RHD deletion become common? Which historical bottlenecks and migrations amplified it? And how did those processes intersect with early human societies and their movements across the globe? To get at those questions, we need to look at what Rh negative meant in the one context where it definitely wasn't neutral: pregnancy. And that's where the story turns from quiet population math to very immediate, very personal stakes. For most of human history, nobody knew what Ra was. But they knew something was wrong. Imagine a woman in a small village, thousands of years before blood typing. Her first pregnancy goes normally. The baby is born healthy. Everyone relaxes. Then the second pregnancy comes, and something changes. The child is born weak, jaundiced, struggling to breathe. Maybe it dies within days. Maybe the third baby is stillborn, swollen with fluid. The mother told it was a curse, a punishment, bad luck, or just how her body is. This pattern haunted certain families. First babies surviving, later babies mysteriously dying. No one could see the thread running through it, a tiny molecular mismatch on the surface of red blood cells. The RHD antigen. Here's what was actually happening. If an Rh negative woman carries an Rh positive fetus, those fetal red blood cells look just a little bit foreign to her immune system. Normally, the placenta keeps maternal and fetal blood mostly separate. But pregnancy and childbirth are messy. Late in pregnancy, during delivery or during events like miscarriage or trauma, small amounts of fetal blood can cross into the mother's circulation. For an Rh negative woman, those Rh positive cells are like a tiny unauthorized border crossing. The first time it happens, her immune system is naive. It sees the D antigen, decides this doesn't belong, and slowly starts building a memory response. This process is called sensitization. She becomes alloimmunized against the D antigen. It doesn't usually cause a crisis in that first pregnancy, because the immune response is ramping up late in the game. The danger comes later, in a subsequent pregnancy with another Rh positive fetus. That same woman's immune system is no longer naive. It's primed. The memory B cells she built after the first exposure wake up and start producing anti-D antibodies in quantity. Those antibodies cross the placenta, enter the fetal bloodstream, and begin doing exactly what they were designed to do: target anything with the D antigen for destruction. The fetus red blood cells become the enemy. This is hemolytic disease of the fetus and newborn, often shortened to HDFN. Hemolytic just means red cells are being broken apart. As those cells are destroyed, the fetus becomes anemic. The heart strains to keep up. The bone marrow and liver go into overdrive trying to replace the lost cells. Fluid starts collecting in tissues and body cavities. In the most severe form, called hydrops fetalis, the fetus can become massively swollen, with heart failure and death before birth. If the baby is born alive, the problems continue. The breakdown products of those destroyed red cells flood the newborn's system, especially bilirubin, a yellow pigment. The liver, still immature, can't keep up. Jaundice deepens from cosmetic yellowing to something far more dangerous, bilirubin crossing into the brain, causing permanent damage, seizures, or death. Before modern treatment, many of these babies simply didn't survive. Those who did could be left with serious neurological problems. To the families living through it, it looked like a curse on the mother's womb. To early doctors, it looked like a tragic mystery. In the 19th and early 20th centuries, clinicians started giving it names: erythroblastosis fetalis, fetal anemia, icterus gravis neonatorum. They noticed the same patterns, certain women repeatedly losing babies late in pregnancy, or delivering severely jaundiced newborns that died soon after birth. They noticed that it was more common in multiparous women, those who'd been pregnant before. They noticed it clustered in certain couples. At the same time, blood transfusion medicine was evolving. The ABO blood groups had been discovered around 1900. Doctors had already learned the hard way that incompatible transfusions could destroy red cells and kill patients. By the 1930s and 40s, some researchers were starting to suspect that whatever was killing these fetuses and newborns might be another kind of blood incompatibility, similar in spirit to ABO, but not the same. Then, in 1940, Karl Landsteiner and Alexander Wiener identified a new blood factor they called Orichi, after experiments with rhesus monkeys. The naming history is messy and the details in animals turned out not to be a perfect match for humans, but the label stuck. Very quickly, other researchers connected the dots. Mothers with antibodies against this Rh factor were having babies with destroyed red blood cells. The mysterious erythroblastosis fetalis wasn't a curse. It was an immune reaction against the fetal D antigen. From there, things moved fast. By the 1950s and 60s, doctors understood the sensitization process, how a first Rh-positive pregnancy could silently prime an Rh-negative mother, and how later Rh-positive fetuses paid the price. They learned that they could sometimes rescue affected newborns with emergency exchange transfusions, literally draining out the antibody-coated blood and replacing it with compatible cells. It was heroic, risky medicine. It saved some lives. It didn't prevent the disease from starting. The real revolution came from flipping the problem on its head. Instead of just treating sensitized mothers and damaged babies, what if you could stop sensitization from happening at all? The answer was counterintuitive. Give the Rh-negative mother a carefully prepared dose of antibodies against the D antigen. Not her own antibodies, but someone else's purified from donor plasma. This product became known as Rhode Immune Globulin, or RHIE, marketed under names like Rogam. Here's the logic. Right after an Rh-positive baby is born, some of its cells inevitably leak into the mother's bloodstream. If you flood her system quickly with anti-D antibodies from the outside, those donor antibodies grab and clear the fetal cells before her own immune system really sees them. No clear signal, no lasting memory, no sensitization. It's like shredding the incriminating documents before the immune police arrive. Early trials in the 1960s were dramatic. In populations where RAG was systematically given to Rh-negative mothers after delivering Rh-positive babies, the rate of new sensitization plummeted. Later, doctors realized they should also give RIG during pregnancy if there was any risk of fetal blood mixing after certain procedures, after trauma, and routinely around the third trimester. Each refinement pushed the risk lower. Within a generation, in countries with access to this prophylaxis, hemolytic disease from RHD incompatibility went from a common, often lethal problem to a rare, mostly preventable one. The same families that once lost child after child could now expect healthy pregnancies. But the memory of those losses didn't vanish. In many cultures, stories persisted about certain women whose bodies rejected their children or certain bloodlines where babies didn't take. When doctors finally put language to it, your blood type is rare, your blood is different, your body makes antibodies against your baby's blood, those phrases landed on top of centuries of grief and superstition. Being Rh negative in that context wasn't just a line in a lab report. It was tied to risk, to tragedy, to the idea that a woman's blood could be fundamentally out of sync with her own offspring. That emotional and social weight helped fuel the sense that Rh negative people were somehow set apart, medically and maybe even biologically. Once you have a trait that can kill babies in very specific pairings of parents, that trait stops being an abstract allele and becomes a story. And stories about dangerous or special blood don't just stay in the clinic. They leak outward into myth, into identity, and eventually into the much stranger claims people now make about where Rh negative blood really came from. By the time RHD was linked to hemolytic disease, medicine had already been burned by blood more than once. In the early days of transfusions, doctors thought blood was blood. You had a patient bleeding out. You had another person with blood to spare. Hook them up, or pour one into the other, and hope for the best. Sometimes it worked. Sometimes the patient revived, colour returning to their face, pulse strengthening. Other times it was a horror show. Shivering, chest pain, dark urine, kidney failure, death. Same species, same colour blood, wildly different outcomes. Those disasters pushed people like Karl Landsteiner to ask a simple but radical question. What if humans don't all have the same kind of blood? Around 1900, Landsteiner discovered the ABO groups by mixing blood from different people in test tubes and watching some mixtures clump and some stay smooth. That clumping, or agglutination, was the immune system reacting to foreign antigens on red cells. Once abo typing and cross-matching became standard, blood transfusion got a lot safer. But not safe. Clinicians kept seeing unexplained reactions. Two people both typed as, say, Group O, no obvious mismatch. Cross-match looked okay by the crude methods of the time. And yet, every so often the recipient's red cells were still destroyed. Something else was hiding in those cells, something abo typing didn't catch. In parallel, obstetricians were staring at their own unsolved puzzle, erythroblastosis fetalis. They didn't know it was the same problem yet, but they were all bumping into the same invisible wall. Some mothers developed strange antibodies. Some neonatal transfusions went horribly wrong even when ABO matched. The clues were scattered across specialties, journals, and languages. The breakthrough that gave Rh its name didn't start with human tragedy. It started with animal experiments. In the late 1930s, Landsteiner teamed up with Alexander Wiener in New York. They did what many immunologists did at the time: immunize animals with foreign red cells and see what antibodies appeared. One of their test subjects was the rhesus macaque, a small monkey that had already become lab workhorse material. They took red cells from a rhesus monkey and injected them into rabbits and guinea pigs. Those animals' immune systems, doing what immune systems do, produced antibodies against the monkey cells. Then Landsteiner and Wiener took those anti-rhesus antibodies and tested them against human blood. To their surprise, the antibodies reacted with the red cells of most humans, but not all. Some people's cells were clumped by these anti-rhesus antibodies. Others weren't. That meant there was a human antigen, present in some individuals and absent in others that looked similar enough to something on rhesus monkey cells to fool the immune system. They called that human factor Rh, after the rhesus monkey. In their original framing, people whose red cells reacted with the rabbit serum were Rh positive. Those whose cells didn't were Rh negative. This is where the story immediately gets messy. The thing Landsteiner and Wiener first called Rhe in monkeys is not actually the same molecule as the human D antigen we now obsess over in pregnancy. The animal and human systems turned out to be analogous, not identical. Later work teased apart the details and showed that the rhesus factor in monkeys was a different antigenic system. By then, the name had already escaped the lab. Doctors started testing for Rh in humans. Other researchers, most notably Levine and his colleagues, noticed that mothers who produced antibodies against this new factor were the same mothers whose babies were dying of erythroblastosis fetalis. The dots connected quickly. Mothers who were Rh negative, fathers who were Rh positive, babies who inherited the father's Rh and triggered the mother's immune system. The clinical link was so strong that, within a few years, Rh was no longer a curiosity. It was a life and death blood group system, second only to Abo in importance. But the naming confusion never really went away. Immunohematology, the field that studies blood group antigens and antibodies, exploded in complexity. Under the Rh umbrella, researchers found not one antigen but many D, C, C, E, E, and a whole constellation of less common variants. D was the big one, the one that caused the most severe reactions and the classic hemolytic disease of the newborn. But strictly speaking, Rh refers to an entire antigen system encoded by a set of related genes, not a single switch. Clinically, though, people simplified. You were either Ra positive or Rh negative, based almost entirely on whether you had the D antigen. This shortcut made sense in hospitals. D is powerful, and D incompatibility was the obvious obstetric killer. But it fused a complicated antigen network, a messy history of monkey experiments, and a specific human protein into one blunt label, Rh. That label felt scientific and exotic at the same time. Rhesus factor, rhesus negative. You can almost feel the distance from everyday life in the word itself. Not type A or type B, which sound like letters on a filing cabinet. Rhesus, an animal name, a lab animal, something not quite human. It didn't help that the early literature sometimes loosely equated RHH with monkey antigen before the distinctions were understood. From there, misunderstanding was almost guaranteed. In modern immunohematology, Rh doesn't mean you have monkey blood or you're missing a monkey gene. It doesn't imply primate ancestry any more than Abo implies you're part of some A-blooded subspecies. All humans have primate ancestry. The Rh system is just one of many blood group systems that diversified as our species did. What Raj does mean is this: lurking in the membrane of your red blood cells, you either express a set of proteins called RhD and RhCe in particular combinations, or you don't. If you lack functional RhD protein, you're classified as Rh negative for transfusion and pregnancy purposes. If you have it, you're Rh positive. That's it. No secret lineage encoded in the name, no direct tie to rhesus monkeys beyond a historical accident in nomenclature. But outside the lab, names don't stay technical. They soak up emotion and story. Doctors trying to explain complex immunology to patients boiled it down for speed and clarity. You have a rare factor missing. Your blood doesn't have the Rh protein. Your blood is different from most people's. Over time, those simplifications detached from the nuance. Missing quietly shifted from lacking this particular antigen to missing something humans are supposed to have. Add in the monkey reference, and suddenly the door is wide open. If Rh positive people are more like monkeys, are Rh negative people less like them? More human, less human, other, that's not what the science says. But it is how human storytelling works. Any time biology gets complicated, we flatten it to something our brains can hold in a sentence. Survival of the fittest. Junk DNA. You only use 10% of your brain. Rh negative blood is missing the rhesus gene. These aren't precise descriptions, they're narrative hooks. And once a hook is in place, myths grow around it. The Rh system is a perfect candidate. It's invisible to everyday experience, ferociously technical when you dig into it, and yet it has direct consequences for sex, reproduction, and survival of offspring. The name came from monkeys, the effect shows up in babies, and the explanation sounds like a riddle. Your body can attack your own child because of a protein you don't have. Out of that tangle of lab mistakes, naming accidents, and clinical shortcuts. A simple idea took root in the public imagination. There is a special blood factor linked to monkeys that some people lack. And whenever science leaves a gap between the technical truth and the simplified story, folklore rushes in to fill it. The Rh negative Enigma wasn't born in a UFO forum or an esoteric blog. It started in glass tubes, monkey cages, and delivery rooms. The myths came later, riding on a single sticky word that no longer means what people think it does. If Rh is just another blood group system, you might expect it to be scattered more or less evenly around the world. A simple on-off switch, half the planet this way, half that way. But that's not what we see when we actually count. When blood banks and epidemiologists started mapping RhD around the mid-20th century, a pattern jumped out immediately. Rh negative isn't sprinkled uniformly like sugar on a donut. It clumps. In many parts of East Asia, more than 99 out of 100 people are Rh positive. Rh negative is down at 1%, sometimes lower. In sub-Saharan Africa, the numbers float in the low single digits. You can find Rh negative individuals, but they're rare. Move into the Middle East and South Asia, and the frequency climbs a bit into the few percent range. Then you look at Europe, and suddenly the graph spikes. In much of Western and Central Europe, roughly 15% of people are Rh negative. That's about 1 in 6. In some populations it's closer to 1 in 3. And in a few small, historically isolated groups, higher still. This is the first hook for a lot of the Rh negative mythology, those European peaks. If a trait that can cause lethal pregnancy complications is missing in only a small slice of humanity, that slice must be special, right? Chosen, ancient, different. The reality is both less mystical and more interesting. To understand why some regions have lots of Rh negative and others almost none, you have to think in terms evolution actually uses: founders, drift, and isolation. Imagine a small group of people moves into a new valley 10,000 years ago. Just by chance, a couple of them are Rh negative. In the big ancestral population, Rh negative might only have been, say, 5%. But in this tiny band, maybe 2 out of 10 founders carry the RhD null version. Suddenly, your starting frequency in the valley is 20% instead of 5. Now lock that valley off, not with steel walls, but with geography and culture. Mountains, dense forests, language barriers, endogamy, war with the neighbours. The people there mostly marry each other, generation after generation. The Rh alleles they brought in at the beginning get recycled inside the group. No new genes drift in from outside, very few drift out. Under those conditions, random chance alone, genetic drift, can amplify whatever frequencies you started with. Sometimes a rare variant disappears. Sometimes it gets lucky and becomes common. Over hundreds of generations, you end up with a local population whose genetic profile looks weird compared to global averages. Not because anything supernatural happened, but because small numbers and isolation magnify quirks. This isn't unique to Rh. You see the same logic in the high rate of certain metabolic diseases in the Amish, or specific inherited disorders in small island populations, or eye and hair colour patterns in Northern Europe. A few founders, a closed network, and enough time can turn a background variant into a defining regional feature. Rh negative appears to have ridden that process in several European lineages. Which brings us to the Basques. If you've ever fallen down a late-night rabbit hole on Rh negative, you've seen the Basque claim. The story usually goes like this. The Basques, tucked between Spain and France, speak a language unrelated to anything else in Europe. They have an unusually high rate of Rh negative blood. Therefore, they are the original Europeans, or survivors of Atlantis, or carriers of a pre-human bloodline, or descendants of whoever supposedly added the code that shouldn't be in us. Strip away the myth layer, and a few solid facts remain. Basque-speaking populations really do have high Rh negative frequencies, often quoted around 25 to 30%. That's significantly above the European average. Their language, Euskara, is a linguistic isolate, not obviously related to Indo-European tongues like Spanish or French. Genetic studies also show some continuity between Basque populations and ancient hunter-gatherers and early farmers of Western Europe, suggesting deep regional roots. But none of that requires aliens or Nephilim or a separate human species. What it does require is what we already talked about. A relatively small ancestral group, geographic refuges in the Western Pyrenees, cultural boundaries that limited intermarriage with neighbours for long periods, and a lot of time. Under those conditions, traits like Rh negative can climb and stick, and the Basques aren't unique. Other groups held up in fringe Rh negative lore, some Celtic language populations, a few pockets in the British Isles. Certain mountain communities also show elevated Rh negative frequencies compared to the global average. In each case, when you pull up the rest of their genetic profile, you don't see a new species. You see perfectly ordinary Homo sapiens, with regional quirks shaped by founders, drift, and partial isolation. The temptation is to read too much into borders on a modern map. Spain has X% Rh negative. Ireland has Y%. Japan has Z, but countries are recent. Blood group alleles don't care where the passport office draws a line. When you say the French are such and such percent Rh negative, you're averaging together descendants of Ice Age hunter-gatherers, early Near Eastern farmers, steppe pastoralists, Roman soldiers, medieval traders, and recent immigrants from every continent. The same is true, in different proportions, for almost any modern nation. Trying to match today's national categories directly to ancient peoples is like trying to guess the recipe of a soup after it's been simmering for 5,000 years with new ingredients tossed in every few centuries. You can spot some lingering flavours, but you can't pretend it's the same as the original broth. This is where Rh maps become both hypnotic and misleading. You've probably seen them, blue here, red there, colour gradients washing across continents. They look authoritative. They whisper, "Here is the truth, encoded in percentages." But those gradients flatten a messy reality. They don't show sample sizes. They don't show city versus countryside, or one valley versus the next. They don't show historical population replacements, plagues, conquests, or migrations. A bold red patch labelled 30% Rh negative might be based on a handful of villages tested in the 1950s. A pale region might simply be undersampled. On top of that, the raw number is only one dimension. Saying Group A is 20% Rh negative, Group B is 5% sounds dramatic. In evolutionary terms, it's just a difference in allele frequencies that could have arisen from a mix of drift, founder effects, and ordinary demographic history. No secret upgrade, no missing human component. None of this makes the distribution of Rh negative boring. If anything, it makes it more powerful as a clue. Those regional spikes and troughs are signposts pointing to past bottlenecks, migrations and isolation events in our species' story. They tell us that whatever gave rise to the RHD negative variants probably happened once or a few times, then was carried, shuffled and amplified by the same forces that shaped everything else about us. Where people go wrong is in turning those signposts into borders between kinds of humans. High Rh negative frequency doesn't mark an enclave of non-humans or a preserved pre-Adamic lineage or a hidden royalty of blood. It marks a population history that tilted the odds for a particular allele. That's still a puzzle worth solving. But to solve it, you have to zoom in deeper than national maps and colour gradients, down to genes, ancient DNA, and the actual origins of the Rh system itself. Which is where we're going next, into the question that haunts almost every Rh negative discussion online. If RHD is the normal human state, what does it really mean genetically and evolutionarily to be missing it? When people online say Rh negative means you're missing a gene, it sounds dramatic, like you've got a blank spot in your DNA where something important should be. A gap, an absence, maybe even a smoking gun for outside interference. But that's not how genetics works. Start with the basic layout. On chromosome 1, there's a stretch of DNA that encodes proteins we call RHD and RHCE. They're cousins. They sit next to each other in the genome. They probably arose by an ancient gene duplication long before humans existed, and then diverged into slightly different versions with slightly different jobs. Most people on Earth carry 2 working copies of RHD, one from each parent. They make the RHD protein, it goes on their red blood cells, and we label them Rh positive. So what does Rh negative mean at the molecular level? It doesn't mean there's a clean, cartoon-like hole where the entire RHD should be. It means that, for that particular person, the route from DNA to functional RHD protein got disrupted somewhere along the way. And evolution has dozens of ways to break a gene without resorting to magic. The most obvious route is deletion. A chunk of DNA gets snipped out during replication or recombination. Sometimes it's a few letters, sometimes it's the whole RHD coding region. If both your chromosome 1s carry a deleted or disrupted RHD, you don't make any normal RHD protein. Blood tests read you as RHD negative. That's one mechanism. But it's not the only one, because RHD and RHCE are so similar they're prone to a process called gene conversion. During meiosis, when sperm and eggs are being made and chromosomes pair up, the cell's repair machinery can get confused and copy-paste a bit of RHCE sequence over a bit of RHD or vice versa. That mosaic swapping can scramble the RHD coding sequence enough that the resulting protein doesn't fold or insert properly in the membrane. Again, functionally negative. Then you have variant alleles, DNA changes that don't erase the gene but alter it. A single base change can swap one amino acid for another at a crucial site. A small insertion or deletion can shift the reading frame. The gene is still there on the chromosome, but the protein it encodes is altered, truncated, unstable, or no longer recognized by standard blood typing reagents. From the perspective of a blood bank, those people are Rh negative, or weak D, or partial D, depending on exactly how impaired the protein is. From the perspective of internet mythology, they are missing something. From the perspective of molecular biology, they are carrying one of many perfectly ordinary variants in a duplicated, recombination-prone gene cluster. And that last part is important. There isn't just one way to be Rh negative. When researchers sequence the RHD region in different populations, they don't find a single Rh negative gene that's spread worldwide. They find a whole catalogue of RHD null alleles, full deletions, hybrid genes formed by swapping pieces with RHCE, nonsense mutations that introduce early stop codons, splice site changes that mess up RNA processing. Some of these variants are common in Europe, others are more frequent in Africa or Asia. Some are rare and local, showing up in just a few families. In other words, Rh negative is a shared blood group label that can be reached by multiple genetic paths. That alone should cool down the extraterrestrial speculation. If an outside intelligence came along and took out a human gene, you'd expect a distinctive, unusual footprint, the same engineered change, or at least the same precise breakpoint, showing up wherever the trait appears. Instead, what we see looks exactly like what you'd predict from normal mutation processes acting on a gene that sits in a recombination hot neighborhood. So why does the missing gene framing sound so compelling to people? Partly because deletions feel special. We instinctively think of evolution as adding features, new genes, new traits, not subtracting them. But in real genomes, loss is just as common as gain. There are entire human populations with segmental deletions that remove the CCR5 receptor, which HIV uses to enter cells. People with the deletion are resistant to certain strains of HIV. Nobody insists they must be alien. Lactase persistence in adults. Your ability to digest milk is controlled by regulatory changes that keep an old gene switched on, while other regulatory regions have been lost or shut down. Color vision in many mammals has been reduced compared to their ancestors through gene loss. Nature deletes things all the time. A missing or altered RHD is unusual only in the sense that we noticed it because of transfusion medicine. It's not a smoking gun, it's just another entry in evolution's long list of edits. Geneticists can even take a stab at how long these edits have been around. They don't do this by staring at one DNA sequence and guessing. They look at patterns of variation around the mutation, how much the neighbouring DNA has been shuffled by recombination and eroded by new mutations. Mutations that arose recently tend to sit in long, intact blocks of similar sequence because there hasn't been time for many crossovers to chop them up. Older variants are embedded in more scrambled backgrounds. Using these and related methods, researchers can estimate the age of specific RHD null alleles in different regions. Some of the European dominant deletions look to be tens of thousands of years old, comfortably older than agriculture, but younger than the split between major continental populations. Others, especially rarer variants, are much younger, probably arising in the last few thousand years. But these are estimates, not carved in stone dates. They come with confidence intervals and assumptions about population size, recombination rates, and demographic history. Change those, and the calendar shifts. The point isn't that we can say this Rh negative allele appeared on a Tuesday 38,500 years ago. The point is that the data fit ordinary human timescales and ordinary mutational mechanisms. Nothing looks impossibly ancient or absurdly synchronized, the way an engineered, one-time global deletion might. What about going further back with ancient DNA? Can we pull a Neanderthal femur out of a cave, sequence it, and label that individual Rh positive or negative? Sometimes. But there are limits. Ancient DNA is fragmented and damaged. We don't always recover the entire RHD region in good enough shape to see precisely which alleles were present. When we do, we still have to interpret them against modern references. If we see a familiar deletion or a known null variant, we can infer an RHD negative phenotype with some confidence. If we see an intact-looking RHD, we tentatively call it positive. But subtle variants, weak D forms, or entirely extinct alleles can be harder to classify. On top of that, ancient DNA samples are sparse and biased. We have genomes from a handful of Neanderthals, a few Denisovans, and a growing but still limited set of early Homo sapiens from different regions and eras. They're incredible windows into the past, but they're not a full census. If one Neanderthal turns out to carry an RHD negative allele, that doesn't mean Neanderthals were Rh negative. It means at least one individual, in one place and time, had a version that would likely type negative by modern standards. What ancient DNA can do, and is starting to do, is show us that the building blocks of Rh variation are old. Some of the structural quirks in the RHD-RHCE region predate the split between Neanderthals and modern humans. That's exactly what you'd expect for a gene family produced by ancient duplication and ongoing recombination. It pushes the story deeper into our shared hominin past. It does not reveal a neat line where normal humans end and mysterious Rh negatives begin. So when you hear the Rh gene is missing that can't happen naturally, remember what's actually going on. We're looking at a recombination-prone gene cluster with a history of duplication. We're seeing deletions, conversions, and point mutations that knock one member of that cluster offline in different ways, in different populations, at different times. The mystery isn't that something shouldn't be in us. It's that people assume normal evolutionary processes can't produce patterns that feel, at first glance, too sharp, too uneven, too loaded with story. Once you accept how messy and flexible genomes really are, a new question comes into focus. If the genetic mechanisms behind Rh negative are ordinary, how did those altered alleles travel through deep time, through migrations, extinctions, and cultural upheavals to end up concentrated where they are today? To follow that trail, you have to leave the double helix and start walking ancient roads. When you zoom out from the gene and look at the map, something becomes obvious very quickly. Rh negative isn't floating in a vacuum. It's riding on the backs of people. And people do not stay put for most of human history. Europe wasn't a stable stage with Celtic or Basque or Germanic actors moving around on top of it. It was a conveyor belt of populations, waves of movement and replacement and mixing that went on for tens of thousands of years. Start with what we now know from ancient DNA. The first modern humans to move into Europe more than 40,000 years ago weren't the ancestors of most Europeans alive today. They were early pioneers interbreeding with Neanderthals, adapting to Ice Age habitats, and then, in many cases, disappearing as climate swung and competitors arrived. Then, around the end of the last glacial maximum, a new wave spread out, hunter-gatherer groups with their own distinct genetic signatures. They repopulated territories that had been under ice or tundra for thousands of years. For a while, they dominated the continent, but they wouldn't be the last. About 9,000 years ago, farmers from Anatolia and the Near East began moving into southeastern Europe. They brought domesticated plants and animals, new tools, new ways of organizing life. Over centuries they expanded north and west, often intermarrying with local hunter-gatherers, sometimes replacing them outright. This wasn't one peaceful march. It was a messy, uneven process that played out differently in different valleys and coastlines. In some places, farmers and foragers coexisted for generations. In others, one way of life quickly pushed out the other. But by the time agriculture had taken firm hold, the genetic landscape of Europe was already radically different from what it had been a few thousand years before. Then came yet another major shift. Around 5,000 to 4,000 years ago, people associated with steppe herder cultures from north of the Black and Caspian Seas began moving westward and southward. They carried different genetic lineages, different burial customs, and probably new languages that would become the backbone of most Indo-European tongues. Again, the pattern wasn't simple replacement. It was blending, stratified mixing, and regional variation. In some areas, steppe ancestry became dominant. In others, it remained a minority component layered on top of older farmer and hunter-gatherer roots. By the Bronze Age, the average European genome was already a three-way mosaic: ancient forager, Anatolian farmer, steppe herder. And that's before you add in later movements: Phoenician traders, Greek colonists, Roman legions, Germanic migrations, Slavic expansions, Viking raids, Moorish rule in Iberia, and all the subtle, undocumented drifts of people marrying across hills, rivers, and language borders. Now put Rh negative back into that picture. Whatever specific RhD null alleles were present in these groups, they weren't travelling in isolation. They were tied to whole genomes embedded in these shifting mosaics. A deletion common in one ancestral population could become more frequent if that population expanded, or less frequent if it shrank, or was diluted by newcomers. A rare variant could suddenly become more visible if a small group carrying it experienced a local boom or founded an isolated community. This is what population geneticists mean when they say that the distribution of a trait today is the end result of countless demographic events. You can't point at the modern Basque country, see a high frequency of Rh negative, and assume a single pristine origin. You're looking at the outcome of repeated mixing, bottlenecks, founder effects, and chance. But our storytelling instincts hate that. We want one lineage, one people, one moment when the gene appeared and then flowed intact through a chosen bloodline. It's the same psychological pull that makes family legends so sticky. We're descended from a lost prince. Our line goes back to the builders of X. Our blood carries the memory of Y. That desire for a clean, heroic origin story maps neatly onto genetic traits that feel mysterious or rare. Rh negative is tailor-made for that temptation. It's discrete, easy to test, and unevenly distributed. It clusters in some regions and is scarce in others. That pattern begs for narrative. There must be a tribe, a clan, an ancient people who were the original carriers. But once you understand how many different RhD null alleles exist, and how many times human populations have merged and resorted, the idea of a single founding Rh negative race doesn't hold up. You can still have local histories, specific variants that arose in particular groups and spread with them, but they're threads, not a solitary cable. This is where another confusion creeps in. We tend to blur ancestry, culture, language, and genetics into one big category called who we are. They're not the same. Ancestry is about biological descent. Which actual individuals in the past contributed DNA to you? Culture is about learned behaviours, customs, beliefs, technologies, art. Language is a tool for communication. It can spread quickly across genetic boundaries or splinter within a single population. Genetics tracks the movement of genes, which may or may not match where a culture or language line is drawn on a map. You can be genetically similar to people who speak a completely different language and practice a completely different religion. You can share a language and culture with people whose ancestors came from very different places. Borders, ethnic labels, and self-identities shift faster than gene frequencies. So when someone says Ra negative comes from the X people, you have to ask, do they mean a cultural group, a linguistic group, or a genetic cluster? And which time slice are they talking about? The farmers of 6000 BCE? The tribes of the Roman era? The kingdoms of the Middle Ages? Those are different populations, layered on top of each other in the same geography. Against this backdrop, it's not surprising that myths of special blood show up again and again. Ancient Egypt framed royal blood as literally different, a divine substance inherited through specific lineages. Medieval and early modern Europe obsessed over blue blood, noble families maintaining purity through careful marriages. In parts of Mesoamerica, priestly and ruling lineages claimed descent from gods or culture heroes, with rituals reinforcing the idea that their blood carried unique power. East Asian courts kept meticulous genealogies, emphasizing unbroken descent from legendary founders. These stories aren't about ABO or RH groups, they're about social power. If you can convince people that leadership is written into the blood, you don't have to justify it in other ways. If you can mark a subset of the population as biologically distinct, you can create clear in-groups and out-groups. And if you're on the top rung, You have every incentive to treat your own ancestry as sacred and everyone else's as ordinary. It's easy, looking back, to caricature these ancient peoples, superstitious, obsessed with purity, inventing myths to prop up hierarchies. But that's too simple, and it's dangerous in its own way. The same cognitive machinery that produced those myths is still running in us. We too like crisp boundaries, us and them, pure and mixed, chosen and ordinary. We too are tempted to map a complex web of ancestry and culture onto a single trait and declare it meaningful. We just swap in DNA tests and blood types where past societies used omens and divine genealogies. Talking carefully about ancient migrations means resisting the urge to flatten people into symbols. Hunter-gatherers weren't primitive placeholders waiting to be replaced by farmers. They were diverse societies with their own technologies and languages. Steppe herders weren't a monolithic invading horde. They were multiple groups over centuries, some violent, some peaceful, some absorbed, some dominant. Early farmers weren't a single civilization, but countless villages with different customs and fates. And any of them could have carried an RHD deletion. Any of them could have contributed a piece of the Rh negative puzzle to later populations. When you keep that complexity in view, bloodline stories start to look less like revelation and more like selective editing. They pick one thread out of a tangled tapestry and hold it up as the whole picture. The real challenge, and the more interesting one, is to trace Rh negative through that tangled tapestry without turning it into a banner for one imagined lineage. In the next section, we'll see how one particular region of Europe, with its own deep and complicated history, became ground zero for the most famous Rh negative origin myth of all. When people go hunting for an original Rh negative population, they almost always end up in the same place: the Basque country. That mountainous corner of Western Europe straddling what is now northern Spain and southwestern France. Green hills, deep valleys, Atlantic storms, and, if you believe 1 million internet posts, the last refuge of an ancient people older than Europe itself. So how did the Basques end up at the centre of Rh negative lore? Part of it is numbers. Early blood bank surveys in the 20th century noticed something striking. Rh negative frequencies in Basque populations were higher than in most of Europe. Depending on the sample, you can see around 25–30% of Basque individuals testing Rh negative, compared to, say, 15–16% in many other Western European groups, and much lower in most of the world. Once those numbers made it into popular books and articles, the leap was almost automatic. High Rh negative percentage? Check. A region with a reputation for being isolated and ancient? Check. Stories about a mysterious language that isn't like its neighbours? Check. Add a sprinkling of Celtic and Atlantean clickbait, and a myth practically writes itself. But to see how the myth diverges from the evidence, you have to separate 3 things that keep getting blurred together: the Basque language, the Basque genetic landscape, and the specific behaviour of a few alleles in that landscape. Start with the language. Euskara, the Basque language, really is unusual in Europe. It's not Indo-European. It doesn't belong to the same big family as Spanish, French, English, German, Russian, Greek, or Hindi. Linguists classify it as a language isolate, meaning that as far as we can tell from surviving evidence, it has no living relatives. That doesn't mean it fell from the sky, or that its speakers are a completely separate species. It means that whatever sister languages it once had are gone, and that the language families we see around it now arrived later. Genetically, modern Basque people look less alien than the language suggests. Ancient DNA from Iberia shows the same broad pattern we see elsewhere in Europe: early hunter-gatherer ancestry, then Anatolian farmer ancestry, then steppe-related ancestry layered in during the Bronze Age. Basque-speaking populations do have some distinctive twists. They tend to show a bit less of the later step input than many neighbouring groups, and a stronger continuity with certain older farmer lineages in that region, but they are not frozen relics of some untouched Paleolithic tribe. They're participants in the same story of mixing and turnover, just with slightly different proportions and some pockets of continuity. Once you understand that, the high-arched negative frequency looks less like a smoking gun and more like a case study in how alleles behave in real populations. Imagine a smallish, somewhat isolated region. Steep terrain, strong local identity, historically limited long-distance marriage and migration compared to flat, open plains. Now imagine that, purely by chance, that region starts out with a higher than average frequency of an RHD null variant. Not because they're Atlanteans, not because they're Nephilim, but because of who happened to be there when certain lineages settled in after the last ice age and the Neolithic waves. Over centuries you add in isolation, mountain valleys, and a distinct culture that make people more likely to marry within a local pool. Bottlenecks, periods of war, famine, disease, or political pressure that shrink the population, so that the surviving lineages carry whatever traits they happen to have. Founders, a few lineages with Rh negative alleles having more descendants, by luck or local circumstance. Run that process for 1,000 years, and a trait that starts out a little high can end up quite high. You haven't invoked alien DNA or secret priest kings. You've just used plain, boring population genetics, drift, structure, and history. That's the piece that gets stripped out in the popular Basque Rh negative story. Instead, you see a very different pattern. First, a kernel of fact. Basques have a high arch negative frequency, and Basque is a language isolate. Then, an interpretive jump. They must be the last pure descendants of the earliest Europeans. Then, a free fall into whatever mythology fits your worldview. For some, that means Atlantis. The Basques become survivors of a lost maritime civilization. Their Rh negative blood, a trace of pre-flood knowledge. For others, especially in certain Christian fringe circles, the Basques and other Rh negative heavy groups get cast as remnants of Nephilim bloodlines, offspring of biblical sons of God and human women. In still other spaces you'll see the language of pure blood, as if Basque Rh negative were an untainted, pre-mixed inheritance that must be preserved. Notice how each of these takes the same data point and slots it into a different script, Atlantean, demonological, or quasi-racial. The science hasn't changed, the gene frequencies haven't changed, only the story wrapped around them has. So how do you keep your footing when the stories go off the rails? You slow down and ask very specific questions. First, what exactly is being claimed? Is the person saying Basques have a higher Rh negative frequency, which is broadly true for several surveys? Or are they saying Basques are a separate race created by X, or Basques are the only original carriers of Rh negative? Those are very different statements. Second, what counts as evidence? Are they citing peer-reviewed studies in population genetics and hematology, or blog posts and self-published books that just quote each other? Can you actually trace their references back to original data? Or does every citation eventually loop to a single sensational source? Third, what are they comparing to what? If someone says Basques are unique, ask, unique compared to whom, and in what metric? Lots of small, relatively isolated populations have elevated frequencies of particular traits. Sickle cell in parts of Africa and the Mediterranean, certain metabolic disorders in Ashkenazi Jews, specific founder mutations in Iceland or Finland. A high local frequency is not the same thing as cosmic uniqueness. Finally, how are they using the word pure? Anytime a claim about genetics leans heavily on purity, primordiality, or older than everyone else, that's a warning flag. Human populations have been mixing, diverging and remixing for hundreds of thousands of years, pure in a deep biological sense, just doesn't exist. The Basque story is fascinating without any of the extras. You have a non-Indo-European language that survived massive cultural shifts, populations that show traces of deep regional continuity amid waves of change. And yes, a relatively high frequency of Rh-negative alleles that tells us something about how genes drift and cluster in real landscapes. You don't need Atlantean engineers or fallen angels to make that interesting. In the next part, we'll see how the same hunger for special blood that clings to the Basque story got supercharged when people started looking at royal and noble lineages, kings, queens, and dynasties framed as custodians of a hidden Rho-negative inheritance. If you zoom out from the Basque valleys and look at the last few thousand years of human history, you see the same pattern popping up over and over again. Powerful families obsessing over who they're allowed to marry. Elaborate rules about good blood and bad blood. Genealogies stretching back not just a few generations, but all the way to mythical founders, gods, or heroes. On the surface, this is about property, titles, and succession. But underneath, it's about something more primal. The belief that certain lineages literally carry something special inside them. A spark, a blessing, or, if you prefer a darker story, a curse. Ancient Egypt framed its pharaohs as living gods, descendants of Ra or Horus. Chinese imperial genealogies linked dynasties to celestial mandates. European monarchs claimed lines back to Trojan princes, biblical patriarchs, or semi-mythical warrior kings. Over and over the message was the same. We're not just people with armies and land. We're biologically different, set apart. That's the psychological ground where Rh-negative conspiracies love to grow. Because once you already believe in special blood, finding a rare blood type looks like finding the physical proof of the old stories. See? Only certain families have this. The royals are keeping it among themselves. The narrative practically writes itself. But to understand what's actually happening, you have to look at how dynasties really operated biologically, not just symbolically. Historically, noble and royal families did intermarry at extreme rates. Partly for alliances, partly because marrying down was seen as dangerous to status. European houses are the clearest example. Habsburgs, Bourbons, Romanovs, Windsors. For centuries, their marriage pools were incredibly small, looping back to the same cousin lines again and again. Genetically, this doesn't create magic, it creates concentration. Whatever alleles are present in that little social bubble, hair colour, face shape, a hereditary disease, a rare blood type, get amplified by what geneticists call inbreeding and founder effects. Instead of mixing with a huge, diverse population, you're shuffling the same deck of cards in a tight circle. Sometimes this just means a cluster of harmless quirks. Sometimes it means trouble. The Habsburg jaw, for instance, that famously protruding lower lip and chin, is a textbook case of traits getting exaggerated by generations of cousin marriage. Haemophilia, the royal disease that ran through Queen Victoria's descendants and devastated Russian and Spanish courts, is another. A single mutation in a single person spread through interlinked royal houses because marriage patterns kept recycling the same genetic variant. None of this required divine bloodlines. It just required small, closed breeding networks. If Rh-negative alleles happen to be a bit more common in certain European aristocratic families, and there are anecdotes and partial records suggesting some clusters, the same logic would apply. You'd expect those alleles to persist and sometimes concentrate within that group, not because they're mystical, but because the group behaves like a tiny, semi-isolated population. But here's where the storytelling kicks in. Rare, poorly understood traits that show up in elites get noticed. They get whispered about. They get reinterpreted through whatever cultural lens is handy. Take porphyria, a metabolic disorder that can cause sensitivity to sunlight, reddish teeth, and episodes of mental disturbance. In some circles, it became part of the folklore around vampire aristocrats. Or look at the Habsburgs again. Their facial features and occasional cognitive issues weren't just medical facts. They were folded into stories about decadent, cursed bloodlines. Haemophilia became proof, in some eyes, that royal blood was fragile and tragic. For others, it was a sign of being different, chosen, even sacrificial. The same condition, two totally different mythic framings. Reich negative fits this pattern perfectly. It's invisible in daily life. It only becomes critical in specific contexts: pregnancy, transfusion, trauma. Historically, those contexts clustered around people with access to doctors, midwives, and written records. So you'd expect wealthy families, including nobles, to have more documented mystery deaths in childbirth, more notes about blood incompatibility, long before anyone knew what an antigen was. Later, when blood typing arrived and some of those families tested as Ra negative, you've got all the ingredients for a modern myth, a rare trait, a history of tragic pregnancies, and a class that already claims to be biologically special. Add the internet, and suddenly you're reading that all European royals are Reich negative, or that only true blue bloods carry the ancient code. Reality is messier and less symmetrical. We don't have comprehensive, peer-reviewed data on the Rh status of every royal and noble lineage. We have scattered medical records, occasional mentions in biographies, and a lot of retroactive guessing. Some royals were Ra negative, many weren't. Some clusters may exist. Others are pure invention. So how do you tell where the archives end and the fan fiction begins? Start with documentation. Real genealogy rests on parish records, legal documents, marriage contracts, birth and death registers, genetic studies that sample actual people. You can trace lines, check dates, cross-reference with independent sources. Internet genealogical fan fiction works very differently. It piles on connections that are emotionally satisfying, not historically plausible. It leaps from "this family had a tragedy in childbirth" to "they must have been Ra negative carriers," then to "they're part of the secret bloodline," all without any blood test, any lab record, any genetic data. It also loves sweeping claims. All presidents descend from King X. All Rh negative people descend from Y. When you see absolute statements about millions of people's ancestry that rest on one or two over-interpreted family trees, you're not in history anymore. You're in myth building. A useful question to ask is, could this claim be falsified? If someone says, "We have hospital records showing Queen so-and-so was Rh negative," in theory you could verify that with access to archives. If someone says, "All true rulers are Ra negative, but it's hidden from us," there's no way to test that. It's built to be unfalsifiable. That's a red flag. Another clue is how the story handles complexity. Real lineages tangle. They bring in outsiders. They have illegitimate children, adoptions, political marriages to unexpected partners. Over centuries, royal blood spills into the general population, and common blood flows back into aristocracy. If a narrative insists on ultra-clean, unbroken lines from ancient priest-kings to modern politicians, without any of that messy human stuff, it's probably more theology than genealogy. When you strip away the romance, noble families are just small populations with weird marriage rules. They're fascinating laboratories for studying how traits, including blood types, move through tightly knit networks. But the biology doesn't support a separate species of blue-blooded humans. That hasn't stopped people from trying to make Rh negative into the latest chapter of the old royal myth. A hidden inheritance guarded by elites, a marker of some older, purer stock. In the next part, we'll follow that impulse as it jumps completely off the historical rails and into deep space, literally, with the claim that Rh negative is a code that shouldn't be in us at all, because it comes from somewhere, or someone not entirely human. Here's how the argument usually starts. If Rh positive and Rh negative people were really meant to be the same species, the story goes their blood wouldn't fight each other. A mother wouldn't produce antibodies that can attack her own baby. Nature wouldn't design something so self-destructive. Therefore, the trait must be foreign, introduced, a glitch in the code, proof that some part of our population carries genes that don't belong to Homo sapiens at all. It sounds compelling, especially if you already like the idea of secret bloodlines. But under the hood, the logic falls apart. Because incompatibility inside a species isn't rare. It's normal. Your immune system's entire job is to tell self from not self and attack anything in the second category. Most of the time, that's viruses, bacteria, parasites. But evolution doesn't work with a perfect blueprint. It tinkers. It reuses old parts, adds new bits, swaps pieces around. The result is a messy immune landscape, where members of the same species can become immunologically incompatible in specific contexts. You see this all over biology. There are plants that can't self-fertilize because their own pollen is recognized and rejected. There are animals where certain mating combinations produce fetuses that trigger immune reactions, or simply fail to implant. Even in humans, blood incompatibilities go way beyond Rh. A bow is the obvious one. A person with type O blood has antibodies against both A and B antigens. Transfuse them with the wrong type, and their immune system will destroy the incoming blood cells. Nobody argues that A and B types are alien because of that. We accept that different versions of the same biological system can collide. Rh is just another layer of that same basic pattern. The D antigen, the main one we're talking about when we say Rh positive or Rh negative, is like a little flag on the surface of red blood cells. If you have it, you're Rh positive. If you lack it, you're Rh negative. People who never express that flag can, under the right circumstances, see it as foreign. Pregnancy is one of those circumstances, but even there, things are more nuanced than the alien code narrative suggests. For a Rh negative mother to become sensitized, fetal blood has to actually mix with hers, usually during birth, miscarriage, trauma, or invasive procedures. Her immune system then builds antibodies against the D antigen. A later pregnancy with a Rh positive fetus can be at risk because those antibodies are already primed and ready. They cross the placenta, tag fetal red cells, and cause hemolytic disease. It's tragic. It's dangerous. Before modern medicine, it killed a lot of babies. But from an evolutionary standpoint, it's not a smoking gun for two different species. It's an unfortunate side effect of three things that are otherwise adaptive: a vigilant immune system, genetic diversity in blood antigens, and tight maternal-fetal connection through the placenta. In other words, incompatibility can arise inside a single, very much human population, just by normal variation in immune markers. You don't need aliens, you just need evolution doing its usual sloppy iterative work. So why do people jump from that to star races and cosmic engineers? Because the stories are already there, waiting. Across cultures you find myths about beings who descend from the sky, mingle with humans and produce hybrid offspring. The Sumerians had the Anunnaki. The Book of Enoch talks about watchers coming down, taking human women and fathering a different kind of child. Indigenous traditions on nearly every continent contain some version of star people, sky ancestors, or teachers who come from above and upgrade humanity. Most of these stories were never about DNA in the modern sense. They were symbolic ways of talking about kingship, priesthood, or sudden leaps in knowledge. But once you live in an age of genetics, it's tempting to retrofit them. So you get a modern synthesis. The Watchers weren't just angels, they were advanced extraterrestrials. The sons of God were a non-human species. The Nephilim weren't just giants, They were hybrids. And somewhere in that mixed lineage, according to the theory, is the Rh negative trait, the tag in our blood that marks who carries the outsider code. Then comes the pattern matching. People notice that Rh negative is globally rare, but not evenly distributed. They notice clusters in specific populations. They map that onto ancient myths, lost civilizations, UFO sightings, whatever narrative they already find compelling. See, they say, this isn't random, it's the fingerprint. But if you want to claim a non-human origin for a trait, you have to ask, what evidence would actually support that? Bare minimum you'd need a genetic sequence that looks clearly non-terrestrial. Something in human DNA that doesn't resemble anything found in other primates, mammals, or life on Earth in general. Not just unusual, but structurally out of place. Following rules unlike terrestrial biology. A distinct, coherent block of genes that can't be explained by known processes like mutation, recombination, selection, or introgression from other archaic humans. Essentially, a segment of code that sits in our genome like a plug-and-play module with no evolutionary trail. A mechanism for how it got there, evidence of engineered insertion, viral like vectors with signatures of design, or abrupt appearance of complex genetic architectures with no precursors. And ideally, independent confirmation. Detection of similar code in something not from Earth, meteorites, hypothetical microbial samples from another world, anything that tells you this piece belongs to that system, not ours. Now compare that to what we actually have for Rh. We can trace the RHD gene and its relatives across primates. We see how deletions, duplications, and regulatory changes create different Rh phenotypes, including the common form of Rh negative that's basically a missing or non-functional RHD gene. We can map its spread across populations using standard population genetics, founder effects, drift, selection, demographic history. There's no alien code block, no bizarre out-of-context sequence, no discontinuity where a completely foreign gene suddenly appears fully formed. The Rh system sits exactly where you'd expect it to sit if it evolved within the messy, branching tree of primate biology. That doesn't mean we know everything about why Rh negative persists at the frequencies it does, or what subtle advantages or disadvantages it might confer in different environments. There are open questions there, but we don't fully understand the selection pressures is very different from this must have come from space. So why does the alien intervention story hold on so tightly? Because it's simple, because it's flattering, and because it gives people a clear identity in a world that often feels anonymous. If you're Rh negative, and you've gone your whole life feeling different, misunderstood, or just statistically rare, being told you carry a code that isn't human can land like a revelation. It turns random chance into destiny. It transforms obstetric risk into cosmic heritage. Instead of being a quirk in your blood, you're a descendant of star visitors, watchers, or ancient engineers. Narratively, that's powerful. It's also elastic. Any new finding can be folded in. If a study finds an odd correlation between Rh status and some trait, it becomes another clue. If no correlation is found, that's because the truth is being suppressed. The story is unfalsifiable by design, which makes it emotionally stable and scientifically useless. But underneath all the mythology, there's a quieter, stranger truth. You don't need aliens to make human biology weird. Evolution, climate catastrophes, genetic bottlenecks, and hundreds of thousands of years of small, scattered populations have already given us a genome full of unlikely stories. The Rh system, with its missing antigens, dangerous pregnancies, and uneven global map, is one chapter in that book. The question is whether we're willing to read it as it is, complex, contingent, still partly unresolved, or whether we need to import star gods and secret codes to make it feel meaningful. In the next section, we'll see how older, scripture-based versions of this same impulse show up in tales of Nephilim and giants. Another way of saying, some of us aren't quite like the others, and using blood to draw the line. When people move from aliens did it to the Bible told us this already, they're usually talking about one word, Nephilim. In English translations, it gets rendered as giants, fallen ones, or just left untranslated like a brand logo you're supposed to recognize on sight. It shows up briefly in Genesis 6, then again in Numbers, and from those short, cryptic appearances, an enormous amount of modern speculation is built. The basic scriptural framework goes like this. In Genesis 6, right before the flood narrative, you get this strange aside. The sons of God saw that the daughters of men were fair, and they took wives of all they chose. The Nephilim were on the earth in those days, and also afterward, when the sons of God came in to the daughters of men, and they bore children to them. Depending on the translation, these children are mighty men, heroes of old, men of renown. Later, in Numbers 13, when Israelite spies scout Canaan, they report, seeing giants, descendants of the Nephilim, they say, we seemed like grasshoppers in our own eyes, and we looked the same to them. Again, translation choices vary. The Hebrew is compact and ambiguous. The imagery is not. From there, later Jewish and Christian writings fill in the gaps. The Book of Enoch, Jubilees, and other Second Temple texts spin out the story of divine or semi-divine beings, watchers, coming down, taking human women, teaching forbidden knowledge, and siring monstrous offspring. These offspring are sometimes explicitly called giants, sometimes described as bloodthirsty tyrants, sometimes as corrupt hybrids who introduce violence and sorcery into the world. Fast forward a couple thousand years and you get a modern remix. The sons of God become a non-human species, angels, yes, but now often reframed as extraterrestrial or extra-dimensional beings. The Nephilim become hybrids, part human, part something else. Their mighty men of old reputation becomes evidence of superior intelligence, psychic ability, or unusual physical traits. At this point, linking Rach negative blood to Nephilim lineage is just one more step. A way of saying, this rare marker in your veins is the leftover signature of that ancient crossing. It feels scriptural. It sounds like you're taking the text seriously, but to get from Genesis to RH factor, you have to make a long series of quiet moves. The first move is translation. Nephilim probably comes from a Hebrew root meaning to fall or to cause to fall. Are they fallen ones? Those who make others fall in battle, a clan name? The Septuagint, the ancient Greek translation of the Hebrew Bible, renders Nephilim as gigantes, which doesn't strictly mean giants at first, it means earthborn descendants of Gaia. Over time, in Greek, and then in Latin and later European languages, gigantes slides toward our modern sense of very large people. So, one translation decision, made thousands of years ago in a different language with different mythic baggage, nudges Nephilim closer to literal giants. Later translators follow suit. English Bibles cement the word giants in the popular imagination. Sermons, children's Bibles, and folklore lock it in. By the time Rh negative theories show up, the ground has already been prepared. Nephilim equals giants equals non-ordinary humans. The second move is interpretation. Ancient texts don't come with footnotes explaining what's metaphor and what's reportage. Pre-modern readers could treat giants literally and symbolically at the same time. Big in size, big in power, big in reputation. A mighty man of renown might be legendary because he was actually tall, or because he was a conqueror, or because epic poetry inflated his story across generations. But modern Nephilim bloodline theories often collapse that complexity. They take the most literal reading, inflate the hybrid angle from later Enochic literature, and then project a continuous traceable genetic line from pre-flood figures to modern human subgroups. all without any documentation because we simply don't have continuous genealogical records running back tens of thousands of years, much less across a global population. The third move is retrofitting physical traits. Once you've decided Nephilim were hybrids, you can start assigning them characteristics. Unusual height, pale skin, strange eyes, enhanced intuition, high IQ, low body temperature, sensitivity to heat or light. Whatever cluster of traits a community already associates with Rh-negative individuals can be mapped backward onto these ancient figures. The problem is, the texts don't give you that level of detail. Genesis doesn't list phenotypes. Enoch doesn't provide blood panels. There is no biblical verse that says, "And their blood lacked the D antigen on the Rh locus." We are taking a sparse, symbolic story about cosmic disorder and moral corruption, and retrofitting it as if it were a clinical description of a recessive blood group. When you step outside the text and into the dirt, the archaeological record, the picture gets even murkier. People love to circulate photos of alleged giant skeletons, massive femurs, skulls the size of bathtubs. Nearly all of those images that go viral online trace back to hoaxes, digital manipulations, or misidentified remains. Archaeologists absolutely do find tall individuals and sometimes populations that are, on average, bigger or more robust than neighbouring groups. But the range is within what we already know human variation can produce, especially when you add good nutrition or specific physical labour. Giant in ancient literature is often just a relative term. If your average male height is 5'3", and you meet a 6'5" warrior wearing metal and carrying a spear, he is a giant to you. The same way a war elephant is a monster, not because it's outside biology, but because it's outside your daily experience. Scientific claims about giants or about a distinct Nephilim subspecies have to clear the same hurdles as any other claim. Replicable skeletal finds, clear dating, anatomical analysis, context within known human variation. So far, there is no established population of hominins that matches the biblical Nephilim narrative and stands apart from the species we already know. Sapiens, Neanderthals, Denisovans and their close cousins. That doesn't mean the stories are worthless. It means they're not lab reports. So how do you respect belief while still being honest about evidence? One way is to recognize that sacred texts operate on multiple levels at once. They encode memory of wars, floods, migrations, encounters with neighboring cultures. They encode theology, ideas about what it means to rebel, to overreach, to mix boundaries that were meant to stay separate. And they encode status, who gets to rule, who counts as of the gods, who is just of men. The Nephilim narrative sits at the crossroads of all three. You can honour that without pretending it secretly contains a technical manual for the RH system. You can say, these stories tell us that ancient people also sensed that not everyone was the same, that some lineages felt different, privileged, dangerous. You can even, if it resonates with you, see Rh-negative blood as part of your own mythic identity, a personal symbol layered onto an older scriptural drama. But once you step into the realm of biology, molecules, alleles, fossil bones, you're in a different kind of conversation. There you have to be willing to let the data constrain the story, not the other way around. Because otherwise, anything rare can be made into a relic of Nephilim blood. Any difference becomes proof of an ancient hybridization, no matter what the genetics say. In the next part, we'll look at how that same impulse, turning a statistical minority into a mystical type, shows up in lists of Rh-negative traits and what actually happens when researchers go looking for measurable differences instead of legendary ones. Once people decide that a blood type carries some kind of hidden lineage, the next step is almost inevitable. They start writing personality profiles. If you've spent any time in Rh-negative forums, you've seen them. long lists of traits that supposedly go with the blood type. High intuition, empathy, sensitivity to light and sound, lower body temperature, different eye colour, strange dreams, feeling like an outsider, a sense of knowing things without being told. Sometimes more exotic claims get added. Electrical sensitivity, paranormal experiences, healing abilities, an affinity for animals, even an aversion to authority. Individually, none of these traits are unusual. Put together in the right frame, they start to feel like a pattern. This is how the story usually goes. You grow up feeling a little out of step. Maybe you're shy, or socially anxious, or just introspective. You notice details other people seem to miss. Loud environments drain you. You get headaches from fluorescent lights. People tell you you're too sensitive. You feel emotions strongly. You're drawn to big questions, weird books, maybe the night sky. Then one day, you stumble on an article. 15 signs you have Rh negative blood. You click. As you read, you feel a shock of recognition. That's me. That's also me. That's basically my whole personality. At the bottom of the article it says something like, if you recognized most of these traits, there's a good chance you're Rh negative. You look up your blood type. It matches. It feels like you've finally found your category. A name for the way you've always been different. From a psychological point of view, what just happened is powerful and completely human. You were presented with a trait list broad enough that a large chunk of the population would see themselves in it. It mixed extremely common experiences. Feeling different as a teenager, being sensitive to criticism, liking animals, with a few more specific ones, unusual reactions to anesthesia, chronic cold hands or feet. You were emotionally primed to notice the hits and gloss over the misses. That's confirmation bias. Our tendency to notice, remember, and repeat the information that fits what we already suspect about ourselves, and quietly ignore the rest. Once you join a community organized around that identity, the effect amplifies. In Rh-negative groups, people trade stories. I've always known what someone is about to say. Streetlights go out when I walk under them. Doctors tell me my blood pressure is weird, but I feel fine. When you read enough of these, your brain starts building a composite image. The Rh-negative person as a specific type. Intuitive, fragile but strong in some unseen way, tuned in to something other people can't feel. The community becomes a filter. Members share articles that support the narrative. New study shows link between blood type and anxiety. Scientists baffled by Rh-negative immune system. Often, when you track those headlines back to the actual studies, the story is much more modest. Tiny effects. Weak correlations. Often not about Rh at all, but about Arbo blood groups. But by the time the study has travelled through blog posts, TikToks and memes, it's proof that science is finally catching up. So what does the research actually say about Rh factor and traits? First, there are solid, uncontroversial medical associations. Rh status matters in pregnancy. It can influence transfusion compatibility. It has some interaction with certain infections. Toxoplasma gondii is a famous example where researchers have looked at personality and reaction time differences among infected people with different Rh statuses. There are a few hints that Rh-negative individuals might have slightly different risks for some conditions, like certain autoimmune diseases or allergies, but these findings are not always consistent across populations. This is where nuance matters. Most blood type research finds, at best, small statistical associations. Not absolute rules, not destiny. A study might find that, on average, Rh-negative people in a specific region score slightly higher or lower on a particular anxiety scale, or respond a bit differently to a specific pathogen. The effect size might be so small that, if you met one person at random, you'd never be able to guess their status from their behaviour. Science also has a bias, but it runs in the opposite direction of the internet. Researchers are cautious, they correct for multiple comparisons. They worry about sample size, measurement error, confounding variables. If they find a weak correlation between Rh status and some trait, they're careful to say, this could be noise, needs replication, might be specific to this population. That's the culture of science at its best, suspicious of its own excitement. Online, that caution evaporates. Tiny correlation in one Czech sample of toxoplasma-infected students becomes Rh-negative people proven to be more psychic. Slightly different average reaction time under one specific lab condition becomes Rh-negative nervous systems are fundamentally alien. The hesitations and caveats that fill the actual papers never make it into the memes. Another problem, multiple testing. If you run enough statistical tests, Rh status versus hundreds of traits, some of them will come out as significant by chance alone. It's like rolling dice until you happen to get 5 sixes in a row. Without careful correction, you'll think you found something profound when you've actually just tripped over randomness. So are there any big, robust, well-replicated personality differences linked to Rh-negative status? As of now, no. There is no widely accepted scientific consensus that Rh-negative people are more intuitive, more empathetic, more psychic, more creative, or more spiritually inclined than Rh-positive people. There is no established Rh-negative profile in psychology the way there are, for example, well-documented differences between morning types and night owls, or between people with and without certain neurodevelopmental conditions. That doesn't mean no differences exist anywhere. It means that if they do, they're small, context-dependent, and easily swamped by everything else that shapes who you are. Upbringing, culture, trauma, nutrition, education, random life events. So how do you, as a curious person who maybe does feel different, navigate all these competing claims? You can use a simple framework. First question, is this a medical claim or a personality claim? If someone says, Rh-negative pregnant women need ROAD immune globulin to prevent hemolytic disease, that's a straightforward medical claim. It's taught in every medical school. There are decades of clinical data behind it. If someone says Rh-negative people are natural empaths who absorb other people's energy, that's a personality claim. It's much harder to test and much easier to project onto. Second question, does the source link to actual studies, and do those studies match the headline? If an article cites research, look at what was actually measured. Was the sample size large? Were the results replicated in different countries? Or is this one quirky dataset? Did the authors themselves say the effect was small or uncertain? When you see words like preliminary, tentative, or needs further study, translate that as don't build a worldview on this yet. Third question, how specific is the trait list? The vaguer and more universally flattering the traits are creative, sensitive, deep thinker, old soul, the more likely you're dealing with a Barnum effect, statements that feel personal but could apply to almost anyone. If a list describes you and also describes half your friends who don't share your blood type, it's not telling you something unique about your biology. It's telling you something about human nature. Fourth question, what's the emotional payoff? Does the trait list offer comfort, status, or a sense of secret specialness? There's nothing wrong with feeling seen or valued. Humans need that. But when a belief is doing a lot of emotional work, healing old wounds, explaining loneliness, offering a club where you finally belong, it becomes harder to question, even when the evidence is thin. The healthiest stance is to hold both truths at once. You can acknowledge that you feel different, that your experiences matter, that patterns in your life are real to you, and you can also admit that those patterns might not be hardwired into your red blood cells, that they might come from personality, history, or even the stories you chose to believe about yourself long before you knew your RH status. Because there is a real mystery here, but it's not that negative people are secretly another species. The mystery is why humans again and again take small biological differences and spin them into sweeping stories of type, destiny, and hidden meaning. Why we're drawn to the idea that some code in our bodies marks us as set apart. In the next section, we'll zoom out from Rh negative specifically and look at the wider ecosystem of internet mythology, how misinformation spreads, why weird claims are so sticky, and how you can explore these ideas with curiosity without getting pulled into a gravity well of nonsense. If you want to understand Rh negative mythology, you have to understand the ecosystem it lives in. The modern internet is basically a factory for turning half-understood facts into epic narratives. It starts with a seed of reality. Arch factor is real. Our incompatibility in pregnancy is real. The distribution of Rh negative blood in different populations is real. A couple of scattered studies hint at unusual associations with infections or immune conditions. None of this is invented. Then something happens in the jump from real to viral. Someone writes a blog post with a dramatic title. Scientists can't explain this blood type inside. They cherry-pick a sentence from a paper about unresolved questions in the evolution of Rh, inflate it into total mystery, then wrap it in speculation about ancient bloodlines, extraterrestrials, or lost civilizations. Another person copies the blog post, drops the cautious parts, adds a few more claims they heard somewhere else, and posts it to a forum. A third person turns it into a video with ominous music and a thumbnail that screams, "The gene that doesn't belong in humans." Now the story has left the domain of science and entered myth. You can see the fingerprints of misinformation all over this process once you know what to look for. First, vague sourcing. Phrases like "scientists say," "experts agree," or "it's been proven" appear constantly, with no names, no journals, no dates. Sometimes a specific name is used, but when you track it down, it's a quote lifted from a completely different context, or from someone who isn't a geneticist at all. Second, misused genetics terms. Words like mutation, anomaly, hybrid, reptilian brain, junk DNA, or non-human sequence get thrown around as if they're all interchangeable. A perfectly ordinary variant becomes a dangerous mutation. A normal difference in frequency between populations becomes evidence of a separate species. A chromosome fusion event, hundreds of thousands of years old, gets rebranded as the code that shouldn't be in us. Third, fake or misleading charts. You've probably seen the maps. Bright red blobs over the Basque region, northern Spain, southern France, labelled Rh negative concentration, with captions like "proof of pre-flood bloodline." Often the numbers are wrong, outdated, or copied from a single unverified source. Or bar graphs that show IQ by blood type with no axis labels, no error bars, no reference to how the data was collected. These images spread fast because they feel like evidence, even when they're just art pretending to be data. Underneath all of this is a simple structural problem. Algorithms reward certainty and shock, not nuance and caveats. A careful title like "What we currently know about Rh negative blood frequency in Europe" will never compete with "This blood type doesn't come from Earth." Platforms are built to maximize clicks, watch time, and emotional engagement. Content that triggers surprise, fear, or a sense of secret knowledge pulls you in and keeps you there. Nuance is bad for that business model. Nuance says, "We're not sure yet. This might be an artifact of sampling. This could disappear when we get better data." There are multiple plausible explanations. None of those phrases make for viral thumbnails. But they're the language of actual science. So what can you do if you're drawn to this topic, but don't want to be misled? You don't need a PhD. You just need a small toolkit. First tool Track the claim back to its source. If a video says, "a recent study proved," look for a link. If there's no link, that's a red flag. If there is, click through. Is it a peer-reviewed paper or a news article about a blog post? If it is a real paper, check the basics. What's the journal? How many people were studied? Is RH factor actually part of the research? Or did someone just search the paper for RH and build a whole story around a single aside? Second tool, check expert consensus. Not just one outlier. Science isn't a single study. It's a pile of studies, many of which disagree at first, slowly converging towards something that holds up. If one small paper hints at something dramatic, strong link between Rh negative and X, ask, have other groups found the same thing? Are review papers or textbooks mentioning it? Are major medical organizations updating guidelines because of it? If not, it might be interesting, but it's not settled in any meaningful sense. Third tool, look for reproducibility. Have the results been replicated with bigger, more diverse samples? Have different teams using different methods found similar patterns? Claims that only appear in one country, one lab, or one YouTube channel are fragile. They may be accidents, biases, or just artifacts of how the data was sliced. Fourth tool, cultivate a healthy prove-it reflex for charts and maps. If a graphic doesn't tell you where the numbers came from, assume it's decoration, not documentation. Real data is messy. It comes with error bars, footnotes, and explanations. If the picture is simple, dramatic, and too perfect, approach it like you would a magician's trick, impressive, but not necessarily what it looks like. Most important, remember that you're allowed to enjoy the mystery without turning it into a doctrine. You can be fascinated by the odd history of Rh-negative blood. You can read about ancient migrations, bottlenecks, and chromosome fusions, and feel that deep, almost spiritual awe at the fact that your cells carry echoes of catastrophes and survivals stretching back nearly 1 million years. You don't have to explain that feeling by deciding that some blood types are more evolved, more spiritual, or more human than others. That's where mythology crosses the line into ideology. Once you start ranking people by blood, you're not just telling stories, you're building hierarchies. And history has shown over and over that blood-based hierarchies get ugly very fast. Racism, eugenics, purity myths, persecution. The specifics change, the pattern doesn't. Responsible curiosity means holding 2 commitments at once. First, follow the questions wherever they lead. Ask weird things. Why does this trait cluster here? Could an ancient bottleneck explain this distribution? What don't we yet understand about Rh biology? Wonder is not the enemy of science, it's the fuel. Second, refuse to let your wonder be weaponized into us versus them. When you feel a narrative tugging you toward chosen people, secret bloodlines, or those other humans are less real, that's your cue to step back. To re-anchor in the basics, we are one species, one tangled family, all carrying the same ancient fusion in chromosome 2, all descended from the same tiny population that almost didn't make it. In the next section, we'll lean into that humility. We'll look at what actually remains unknown about Rh-negative origins, what the most plausible explanations are right now, and how you can sit with unanswered questions without needing to reach for aliens, lost angels, or forbidden codes that don't belong in our DNA. So let's finally put all of this on the table. If you strip away the YouTube thumbnails, the Tumblr threads, the "my grandmother was Basque and I have green eyes, so" anecdotes, what are scientists actually working with when they talk about Rh-negative? There are 3 big pillars that, together, explain most of what we see. First, plain old mutation. The RhD protein, the one you either have Rh positive or largely don't Rh negative, is coded by a gene that, like every gene, is vulnerable to copying errors. Over human history, Multiple loss-of-function mutations have hit that gene. Some of them delete it entirely. Others scramble it so badly it never makes a working protein. Each time that happens in a sperm or egg that goes on to form a person, that's another Rh negative allele entering the population. This isn't exotic. It's the same basic process that gives some people lactose intolerance, different earwax textures, or the inability to smell certain odours. The Rh negative state is not the appearance of a brand new alien code. It's the absence or breakdown of a very old, very ordinary one. Second, genetic drift and founder effects. Once a mutation exists, it doesn't stay at a fixed frequency. In small or isolated populations, random chance can amplify or erase it. A few Rh negative carriers have more children than average. A village bottleneck leaves behind a surviving group where that allele happens to be common. A migrating clan that settles in a valley or on an island just happens by lottery to have a lot of Rh negative members. Over centuries, that randomness leaves patterns, clusters, hotspots. You don't need secret Atlantean refugees to explain why certain mountain communities or historically isolated groups have 15, 20 or 30% Rh negative. You need a handful of carriers, some historical isolation, and enough time for random reproductive luck to do its thing. Third, possible selection, sometimes for, sometimes against. RhD isn't biologically meaningless. It does something in the membrane of red blood cells. That means that, depending on the parasite, virus, or environmental pressure, having it or not having it could matter. We already know one clear selection pressure against mismatch: pregnancy. In a world without modern medicine, an Rh negative woman having multiple pregnancies with Rh positive partners is at increased risk of losing later pregnancies to hemolytic disease. That tends to reduce the number of surviving children in certain mating patterns, which slightly pushes against high Rh negative frequencies in some contexts. On the flip side, some studies hint that Rh negative may offer relative advantages in very specific situations, slightly different susceptibility to certain infections, or altered risk for particular disorders. The data is patchy and often weak, but over tens of thousands of years, even tiny biases can nudge allele frequencies up or down. So, new mutations add Rh negative. Drift reshuffles the deck and occasionally stacks it. Selection trims and tweaks around the edges. Layer human migrations on top, Ice Age refuges, Neolithic farmers, steppe pastoralists, colonial movements, and you get the jagged, uneven global map we see today. That's the strongest evidence-based story we have right now. But there are things we still genuinely don't know. We don't have a precise timeline for when the major Rh negative variants first arose. We can bracket them. They must be at least tens of thousands of years old to reach current frequencies in some regions, and some likely predate the split of certain ancestral populations. But we don't have a single fossilized first Ra negative person, or a neat date like 35,000 years ago in this specific valley. Genes don't come with timestamps that precise. We also don't fully understand the local histories. Why exactly is Rh negative especially common in parts of the Western Pyrenees, but not in other regions with similar isolation? How did particular variants spread through the Near East and then into Europe? Did Bronze Age trade networks matter? Did specific plagues or environmental shocks wipe out lineages that carried more Rh positive or more Rh negative alleles? We can model possibilities. We can say scenario A fits the data a bit better than scenario B. But there is still a lot of room for uncertainty in the fine-grained story. Then there are the rare variants. Most people think of Rh negative as one thing. Either you have the D antigen or you don't. In reality, the Rh system is a whole cluster of antigens with dozens of rare combinations, partial expressions and hybrid alleles, some of which are poorly mapped. In certain African and Asian populations, there are Rh patterns that don't fit neatly into the standard European-derived categories. We know they exist. We don't yet have large, detailed datasets that show exactly how common they are, how they interact with disease risks, or how they arose. This is where mythology tries to rush in. Anywhere there's ambiguity, a story can be inserted. Scientists don't know exactly when Rh negative first appeared, because science admits it has no idea where this blood came from. We haven't sampled enough ancient graves in Region X because this proves there was an erased civilization whose bloodline survives only in... You know how it goes. But notice what the scientific picture actually looks like when you zoom out. It's not one mechanism to rule them all. It's a stack. Mutation is always happening, throwing new variants into the mix. Drift is always acting, especially strongly in small historical populations. Selection is sometimes strong, sometimes subtle, sometimes even flipping direction when the environment changes. Migration is constantly stirring the pot. Hunter-gatherers, farmers, herders, traders, enslaved people, refugees, all carrying their alleles into new gene pools. These mechanisms don't compete with each other, they cooperate. You can have a mutation arise in one person, get lucky through drift in a small clan, be pushed a bit higher by local disease pressures, then be carried into new lands by migration, and then be trimmed back by pregnancy-related selection in certain mating patterns. That kind of layered explanation is far less dramatic than inserted alien code. But it fits what we see not just with Rh, but with hundreds of other human traits: lactase persistence, skin pigmentation, altitude tolerance, malaria resistance. The puzzle pieces are the same. The final picture is just arranged differently in each case. Future research can sharpen that picture. We're still in the early days of ancient DNA for blood group systems. Most high-profile studies so far have focused on ancestry, skin colour, disease genes. As methods improve, researchers are starting to call classic blood group alleles from ancient remains. A wider, more global sampling, more skeletons from Africa, from Central Asia, from the Americas, from ancient islands and highlands could show us when and where Rh negative variants were already present and in what frequencies. Better modern datasets will help too. Many global health studies simply record Rh positive or Rh negative without genotyping the underlying alleles, and many regions are still undersampled. Large representative projects that combine detailed Rh genotyping with ancestry, health outcomes, and local history could reveal subtle selection patterns we currently can't see. None of this is about ruining the enigma. If anything, it makes it richer. Ruck. Negative isn't a glitch that proves we don't belong here. It's a tiny thread in a vast, tangled tapestry of human variation, shaped by volcanoes and famines, by who crossed which mountain passes and who stayed behind, by ancient diseases we've never named. The mystery is not that a particular allele exists, it's that it sits there in your blood as living record of events your ancestors never wrote down. And that brings us to the last reframing. The real story here isn't just about a blood factor. It's about what we do with it. Give humans any biological difference, height, eye colour, blood type, haplogroup, and we will try to turn it into meaning. We'll build myths of origin and destiny. We'll cast ourselves as survivors, chosen ones, outsiders, or guardians of some secret lineage. Rh negative is a case study in that impulse. The same evolutionary processes that likely explain its distribution, mutation, drift, selection, migration, also shape the brains that love patterns, narratives, and belonging. Your urge to see a hidden story in your blood is itself an evolved trait. In the final section, we'll look at how to honour that urge without letting it run away with you, how to explore your own biology, your own ancestry, and yes, your own Rh status, in a way that feeds curiosity and connection instead of hierarchy and fantasy. So let's bring it home. Rach negative isn't a secret badge of an ancient order. It's not proof that you're half angel, half alien, or not from here. It's one variant in a very old blood system, medically important because of pregnancy. Interesting because of its patchy distribution and irresistible to storytellers, because it feels like a hidden marker. You've seen how the myths form. Start with a real pattern. Higher Rh negative frequencies in some regions, strange medical complications in mixed pregnancies, a patchwork of incomplete data. Add human pattern seeking. Why me? Add identity. My family is different. Stir in the internet, where the loudest, most cinematic explanations spread the fastest. Suddenly, a gene that probably arose through ordinary mutation, and Drift is wearing a cape and carrying a prophecy. You don't have to reject the mystery to step away from the prophecy. You can treat Rach negative the way you'd treat a fragment of pottery your grandmother dug up in the garden. It's a clue. It says, something happened here. People lived, loved, migrated, nearly died out, survived. This little shard is what's left. You can be fascinated by that without deciding it proves you're from a lost continent. So if you're Rh negative, or you have Rh negative kids, or a parent who nearly died from a pregnancy complication before Rogam existed, your story is part of this. If your grandparents told you tales about old blood in the family, or Basque lineages, or nobody in our village had the same blood as the city people, that's worth telling too. I'd actually love to hear those stories. Share them in the comments, but do it with a bit of care and humility. Instead of we're the real humans or everyone else is a different species, frame it as here's what my family believed or here's what I've noticed. And I wonder how it fits with the evidence. That tiny shift from declaration to question keeps the conversation open instead of turning it into a hierarchy. Because curiosity is at its best when it's anchored in data. So you're willing to update when new evidence comes in. Anchored in history. So you see how wars, migrations, and epidemics shape genes. Anchored in empathy. So you don't turn a quirk of biochemistry into a ladder with yourself on the top rung. You can absolutely chase the weird questions. Could certain diseases have nudged Rh negative higher in one region? Did a particular migration wave carry a big founder effect? What will ancient DNA from undersampled regions reveal? Those are great lines of inquiry. They don't require fear or superiority, or a story where some people are secretly more pure than others. They just require patience and a willingness to live with, we don't know yet. So here's what I want you to do next. Drop a comment telling me where you're listening from and what time it is for you right now. Are you in the Pyrenees at 2 a.m? Doom-scrolling RH forums? In Brazil on your lunch break? In a hospital parking lot after getting your Rogam shot? Put yourself on the map, and if you have an Rh negative story, medical family legend, or just how you first found out, add that too. Let's build a comment section that's actually useful to other curious people who wander in here from the algorithm. If this deep dive helped you see the Rh Enigma a little differently, if it gave you better questions to ask the next time someone posts a wild bloodline meme, take a second to like the video. It really does tell the algorithm this kind of long-form evidence-first content is worth showing to more people. And if you want more explorations like this, where we take the mystery seriously without losing the plot, make sure you subscribe and turn on notifications so you don't miss what comes next.